Okay, hi, welcome from my side as well. Maybe you remember me from the psychosis talk, but if you weren't there, I introduce myself quickly. So I'm Leon Martin, and working together with Leon Stefanowski in the brain simulation section at Charité. And yeah, today I'm gonna try to give you a quick, not really quick actually, but a first overview of like processing basics or of MRI imaging or basic neuroimaging analysis. So as a disclosure, I'm here definitely not the expert with years of experience, but I try to make it comprehensive and to put concepts into my slides, which I was struggling with and with which I wished to know this at the beginning when I started working with this kind of data. So yeah, please ask any questions if you want to. We have plenty to go through, but if we won't gonna make it this time, then we can catch up a few things in the next session as well, because they are kind of adding up to each other. So it makes sense that we yeah, not overdo and yeah, take a break in the middle. And please ask whenever you feel that something is not clear. Okay, let's jump right into it.
Now we see it.
Okay, yeah, big sorry for that. Totally forgot to share it. I was looking at my slides. Okay, going back to the outline: introduction, MR physics, data structure, modalities, overview of analysis, and different analysis pipelines. So that was this slide, and now we're coming to the invention of MR imaging, and now you see the faces of Raymond Damadian, Peter Mansfield, and Paul Lauterbur. So those two guys, this one and that one, invented the method to recreate an image, and this guy was the inventor of the first human body MR scanner. So Peter Mansfield and Paul Lauterbur got the Nobel Prize of Medicine in 2003, but Raymond didn't get the Nobel Prize. Yeah, there was a debate why he didn't get it. Yeah, but it was a quite yeah, it shaped the scientific methodologies in so many ways, and was the ground foundation for neuroscience or for investigating a living brain of a human that is kind of ethical. Yeah, so why do we talk about MRI analysis at all? So can we not just throw the subject in the scanner, take a nice picture, and then see what happens? This is not the case, because this, as I said, this measure is super indirect, and there's a high variability of different MRI analysis approaches. And there was a nice study that that provided a data set to over 70 different teams. And the question was to analyze the data with regards to 9 hypotheses. And there was a high variability between the analysis pipelines they used, and between the outcome of the significance test, basically. So as you can see here on the left side, the red dots are the fractions of teams that have a positive hypothesis against they decline the hypothesis. So you can see for some hypotheses there's still a kind of 30%, 20 to 30% that declined the hypothesis compared to the other teams which accepted the hypothesis based on their analysis pipe. So there's a high variability, and this is caused by a high amount of flexibility with regards to different tools or analysis compartments. And yeah, as this flexibility was represented by the case that each team chose a different workflow. There was no two teams that had analyzed their data with the same workflow pipeline, and yeah, this was highly influenced. This highly influenced then the result of the hypothesis test. As you can see here below, there were, for one hypothesis, there were different clusters of teams that showed different results based on the same data. So they all got the same data, but based on the analysis procedure, they got different results. So clearly in this cluster, there are so many activations, and in this cluster, there are almost no activations. So this is a rather conservative approach, and this is a more liberal approach. As you can see here, with a really conservative approach, the probability to accept the hypothesis was rather only the half or a third of those approaches which were more liberal. Yeah, and this was depending strongly on the kind of tools they used, the software they used. Okay, so now I hope everyone knows that it is quite interesting and quite important to know what an analysis of magnetic resonance imaging is about. So how we get to the image is another thing. So let's try to answer the question how it is even possible to take a picture from the inside of a living brain. So for me, at least, it's quite hard to intuitively get or invent a method or think about how this would be achieved. So I tried to make it really simple for the first term. So there are different steps. First, the subject is placed in a big magnet, basically. Then a radio wave or radio frequency pulse is sent in to the magnet and turned off again. And based on this radio wave, which introduces energy, the patient emits a signal. So it kind of comes back, and that can be received by the scanner and used for reconstruction of the picture. So, okay, that was the simple step, and now a bit more in depth. I chose to rely on those kind of old-fashioned 70s like pictures, or with the purple, because I liked it a lot. So please bear with me. So all protons possess a certain spin. So protons are electrical charges, basically, and they turn around a certain axis, like the Earth is doing as well. But protons have a charge and they spin. And moving electrical current induces a magnetic field, as we know from physics. So protons can be seen as little bar magnets. And when you place those protons, or a subject that possesses a lot of those protons, because they make up every molecule, if you place then the subject in a big magnet of an MR scanner, the protons are lying with the external field in parallel fashion, like south or north aligned, or in anti-parallel fashion. So the North Pole points to the south. So that's the alignment in an external magnetic field, and that's what the M for MRI stands for. So magnetic, an external field that is applied to the subject, and this is also called B0 field. So the protons are not only aligning and rotating, but they rotate in a cone-like fashion around the magnetic field lines of the external magnetic field, and this is called precession. And the precession actually goes really fast. So the precession frequency for a hydrogen proton, for example, is somewhere around 42 megahertz in a magnetic field strength of 1 Tesla. So you can see this can be calculated with a Lamour equation. So this is the precession frequency, and this is the baseline magnetic field strength. and this is like a constant for different protons, different nuclei. And so we can say that in a field strength of 1 Tesla, a hydrogen nucleus has the precession of 42 megahertz, which is like 42 million times per second. So the stronger the magnetic field, the higher is the precession frequency. If we then introduce like a coordinate system, we see that if we put the patient or the subject in the scanner and all protons align with the external magnetic field, we can describe the locations of the protons with the same coordinate, with the same axis. And each proton is then precessing along the external magnetic field lines. So the subject becomes basically the magnet and develops his own magnetic field along the external B0. This is called longitudinal magnetization. And this cannot be measured directly. So this could be nice because this is what it is used to get a signal, but we have to think about a get around. So why can we measure this? So we can imagine that you're standing in a boat and floating down a river, like in the z-axis, and you have a water hose in your hand and sprinkle water into the river. And for somebody who is watching you from the shore, it is not possible to tell how much water you pour out. And this shall be how much new magnetization is added to the old direction. So you can't see from the shore. But when you point the water hose to the shore, you change the direction of the new magnetic field, then the water may perhaps be directly picked up and measured by an impartial observer, that's standing on the shore, the MR scanner. And so magnetization along or longitudinal to the external magnetic field cannot be measured directly, but when we have a magnetization which is not longitudinal along the river, but transversal to the external magnetic field, we can measure it. So we can pick up a signal of magnetization when we stand next to like the river, basically. So that's the analogy of measuring like transversion magnetization. And this is then the basic fundamental basics for measuring an MRI signal. Then what the R stands for is resonance. So how could we measure this transversal magnetization? Because if we place a subject slowly in the scanner, then we have this longitudinal magnetization along the B0 field, along the external magnetic field. But sending in a radio frequency pulse, which has the same frequency as the proton precesses, then we get two effects. So we know the frequency of protons, how they precess, because we have this Lamour frequency, Lamour equation. And now we can send in this radio frequency pulse at the same frequency. And if it is at the same frequency, protons can pick up energy from this pulse. And this energy transfer is called resonance. So they can pick up the energy if it's the same frequency. So think about like a tuning fork or something, or a piano. If you go into a room with different tuning forks and each have different frequencies, so A, E, so notes, and G, and if you hit one tuning fork with the frequency of A, then only the tuning forks that have the same base frequency will resonate, because they have the same frequency and they can pick up this kind of energy. And the same goes with protons and the radio frequency pulse. Okay, if they pick up energy, they can kind of go somewhere else and don't have to align to the base magnetic field, so they can decrease the amount of longitudinal magnetization. And what the second thing is, what happens, protons get in sync. So if energy comes in, they precess in phase, and if their vectors add up, we have a direction transverse to the original magnetic field. So we have a transverse magnetization that is established. So the radio frequency pulse decreases longitudinal magnetization and establishes transversal magnetization. So a longitudinal decrease and transversal magnetization increase because the vectors add up towards the new axis. This transversal magnetization is also moving around with the precession. And so magnetization that constantly changes its direction, also called an oscillating B1 field, is causing an electrical current. Because if you have a coil and inside the coil is a moving magnetic field, then the coil can pick up, or the antenna in this case, can pick up this moving magnetic field, and an electric current is then induced. And yeah, this is basically then a resulting MR signal, which has exactly the same frequency than the precession. So that is the first approach to get an MR signal. So we get like the frequency of the moving protons that got energy from the radio frequency pulse. So if we now switch off the pulse, the system relaxes, and again the longitudinal magnetization increases. This is also called a longitudinal relaxation time, also called T1, or thermal relaxation, or spin lattice relaxation. This is based because on transfer of thermal energy from the spinning protons that got energy from the radio frequency pulse to the environment. And but they can't keep this energy, so they give it up in heat, basically. And so this is thermal energy. Or they give it to the environment, also called lattice, so spin lattice relaxation. So this looks like this. So we have, as the radio frequency is switched off, and the protons go back align with the longitudinal magnetization. And yeah, at a certain point that is defined with this function, like at 63% of the original magnetization, we can set a time constant basically to measure the speed of the longitudinal relaxation. And this is called T1. At the same time, the transversal magnetization decreases because the spins realign, the protons realign. And this decrease can be described with the transversal relaxation time, T2. And it has to be said that there are different and independent processes. And this is really important because if one-to-one predicts the other one, then we can't differentiate between both, but the processes are independent. And so we can describe this transversal relaxation with another curve. So the T1 with 300 to 2000 milliseconds is way longer than T2 decay. And so it's defined, T1 is defined when 63% of the original longitudinal magnetization is reached, and T2 is defined when transversion magnetization is decreased to 37%. So this said, we can see like the transversal magnetization reducing, and we can take advantage of those processes because different tissues have different relaxation times. And that's basically it. We can measure how fast the protons go back to the original magnetization to the external magnetic field. And this time constant is different for different tissues. So we can differentiate between tissues based on the relaxation properties. So how can we get out an image from that signal? So we coming for the eye of imaging and gradient fields. The MR signal is it's basically made up of a sum of all nuclei inside the scanner. So we have a sum signal, and we have to somehow decompose the signal to separate individual contributions. So to determine where in the head the signal comes from. And this is achieved with applying another external magnetic field, and this is called gradient field. And the magnetization of a gradient field is different for different locations in the brain. So signals from different locations have different frequencies because the external magnetic field strength is different in different areas of the brain. So the frequency content of the received signal can be decomposed regarding different frequencies because we know where the gradient is, and the signal depends on the location. So we can say, okay, this frequency must come from this part of the brain because we applied there the gradient of the x. And so we have for each axis a different gradient. So the last couple of terms regarding to more the physics and engineering side of this, we have first of all the term contrast and echo time. So the MR signal is kind of a measure of net magnetization of those nuclei and those photons. So it has not a direct physiological meaning. So we can have only relative measures of signals of decay or an increase in magnetization. And this measure is due to its relativity, we can think of contrast. So contrast between tissues based on relaxation properties of hydrogen nuclei. So for example, if you to visualize this, if you look at the right, you see two different tissue types, tissue 1 and tissue 2, which have different relaxation properties for the T2 relaxation. So the decrease in transversal magnetization. And now we get a radio frequency pulse, and we can decide when we want to measure the signal. And based on this decision, the echo time, when the signal gets collected, basically, we see that there are differences in signal strength and contrast between both signals. So if we have a short T echo time, TE1, we have high signals, so this can be seen here in a brighter image, but the contrast between both tissue types is not good. So if you have a longer T echo time, then we get a lower signal, but a much higher contrast between the different tissue types. And if we wait a bit longer, then we get a low signal and a low contrast because then there's an asymptote. So we have to think about what we want to measure, so which tissues we want to contrast to each other. And this leads us to a decision of a certain echo time. So how long do we wait to measure the signal? Okay. Another thing is that the time taken between excitation pulses. This fact that T1 relaxation is a lot slower than the T2 decay is used to contrast T1 weighted images. So if we choose a repetition time that is sufficiently short so that the net magnetization aligned with the field does not fully recover, then the next excitation will start with a smaller magnetization, and so the signal we measure will be reduced at our chosen echo time. So this can be seen here, and we receive then a higher contrast with a basically relatively lower signal, but a higher contrast for this T1 weighted image. So T2 weighted image is based on the echo time, T1 weighted image is based on the repetition time, basically. But we can see, we go to the stage later, and maybe it makes sense then more. But for the terms and for this kind of superficial introduction into the physics and hardware, we should take a look at the scanner. So in order to create and measure those magnetic fields, the MR scanner uses a collection of different coils, made of wire, conducting wire. And those coils are used because of two nice features of it. They can, on the one hand, create magnetic fields. So if you pass electric currents through a coil, they can create a magnetic field. So B0, baseline magnetic field, external magnetic field, the gradient field, but also changing magnetic fields with the effect of like the change of transversal and longitudinal magnetization of the protons. If it's inside a coil, it can induce an electrical current in the coil. And so we can measure the magnetization with coils also. This is mainly done by the head coil. So this is a quite nice way of applying and measuring magnetization with those coils. Okay, to sum it up, so hydrogen nuclei in molecules can act as like little tiny bar magnets, and they can be manipulated by magnetic fields when we pass currents to the coils of the MR scanner. So in addition, nuclei produce then like small magnetic fields of their own, and those fields are then sufficient to generate small electrical currents in those coils. Okay, any questions so far? Well, I'm assuming not. Silence is good, I think. And yeah, then we can jump to the MR modality. So I don't know if silence is good, so please go for a little interactive part. It's your turn. So there are like 4 different standard visualization of different MRI modalities. And please, would be somebody so nice to like just name those modalities as a little quiz. This would be super nice. You can also post it in the chat if your microphone doesn't work or something. Okay, which modality is this? Come on, little quiz. Okay, now I'm really thinking if you can hear me at all.
This should be T1.
Yeah.
DTI, so the diffusion tractography.
Yes, it's diffusion weighted image. What's depicted here?
Is it
Okay.
functional?
Yes, it's a functional. It's a statistical analysis of a functional image. Yes, and this is the bonus question. Okay, I wasn't sure at the beginning as well. So it's perfusion MRI. Okay, but I won't talk about this because this is beyond the scope of the talk, so it's totally fine. So thanks for the interactive part. Okay, what is an image basically? So you see the visualizations of the image, but how is this represented in a machine readable fashion? So we have a 3D volume of a brain that corresponds to the real 3D brain in our heads, and yeah, like two-dimensional pictures and pixels, it has like little compartments of three-dimensional cubes called voxels. And there can be different orientations of the voxels. They can have the same edge length in all 3 dimensions. This is called isotropic voxels, and they can have different edge sizes. This is called anisotropic voxels. And for research, mostly we are interested in isotropic voxels. Yeah, then another term for image fundamentals is the resolution. Here you can see a fading between 1 millimeter resolution of an MR scan and the 100 micrometer resolution of the big brain. And yeah, it's clearly obvious what we are dealing with most of the time. We're not dealing with 100 micrometer resolutions in MRI analysis. We're dealing with like in a 1 millimeter dimension. So it's kind of blurry if you know how it should look like, but we can work with it. And another term is time series. So this makes up the 4th dimension. So we have a 3D volume measured over time. So each voxel can be then measured in different time intervals, making up a 4-dimensional image for the 3D volume and the time series. There are 2 main image formats. The one is DICOM, the digital imaging and communications and medicine format, and then there's the nifty file. DICOM is mostly used for clinical use, and has directly stored lots of metadata about acquisition and parameters about so everything is stored in this DICOM. And in the nifty format, this is mainly used in research. And yeah, in a file, only the resolution and orientation is stored. But this is more efficient for imaging process and analysis, and you can store metadata in a separate file. So this is the Neuroimaging Informatics Technology Initiative when they defined data structures for neuroimaging formats. And like I think it's already the 2nd revision of this standard. And how the nifty file looks like then, or how the computer reads in this nifty file, there's a header which stores information about the data type and the orientation. And then there's an image with the signal in each voxel, basically in a 3D array. And this makes up then different slices of the brain, making up a 3D volume. So this is how the information of an image is stored in a nifty file. Because we have only this 3D array of numbers, we have somehow to correspond to the real world coordinate system, basically. Because we have different coordinate systems. We have the coordinate system of the scanner, of the human, so superior, posterior, superior, inferior, anterior, posterior, and left and right. So we have sagittal, coronal, and axial coordinate system, but we have also like the KJI voxel matrix coordinates, and because everything is kind of grid-like, so we can define a transformation matrix that transform between the image array of voxels in the 3 dimensions to real-world dimensions like left and right, superior, inferior. And this is called affine transformation, so a coordinate transform. This is stored in an affine. So we have different rotations and transformations stored in one matrix, and we can multiply our voxel array with this coordinate affine to get the real world transformation, real world coordinates. Yeah, this is a basic matrix multiplication that shifts and rotates the voxel coordinates so that they correspond to the real world coordinates. Yes, so we have left, right, bottom, top, back, front, and yeah, only those coordinates. Okay, I've written a lot in my notes for this slide, but I think this will be sufficient for now. If you have any questions for the affine, it's a bit complex, and but if you once made your head through it, then it makes sense. So it's basically just the transformation between different coordinate systems. So that's it. So we have this numbers stored in an array, and with this affine stored in the Nifty file, we know where each number in the brain is located. That's it, basically. Okay, because there are so many different approaches, and the analysis of MRI data is so flexible. Yeah, there's a data structure of brain imaging data, and this is defined of the BITS consortium to make data reusable and to share data with a common structure. Because as you've seen in the beginning, yeah, 70 teams have 70 different pipelines, and there are so many teams working with neuroimaging data. So the amount of different pipelines and structures is huge. So there is definitely the need for a simple and intuitive way to organize and describe your neuroimaging data. So that's the BITS. It's for efficiency, and there's also software that directly understands the BITS organization. So if you have a data set organized in this structure, you can directly run analysis pipelines with certain softwares, and you don't have to think about which image was what. Is this image already pre-processed? Do I have to do anything with it, or can I do just a statistical analysis, or which reprocessing was applied? This can also all be stored in the BITS format. So we have arranging data and writing down metadata. We have like kind of a definition of compulsory, optional, and additional data for each modality, and we have also automated validation tools that say if our BITS data set is correctly made or not. There are many tools for bitsifying your data sets, so you can convert your raw MRI data in DICOM format to a BITS compatible dataset format, and then just run analysis pipelines with it without thinking of reorganizing your data and how to store it and how to find it again. Yeah, that's pretty nice. And if you want to understand this a bit bumpy data file name structure, it makes a lot of sense, and you can easily find the image you want to find in a data set from another team without always asking them about which file is stored. So we have a chain of entities, which is defined in a key value pair. And then we have a modality, a suffix, and an extension that is defining the data format. Yeah, so we have, for this case, subject and the subject label. Then we have the session and the session label. So the session is kind of a logical grouping of neuroimaging data that is across subjects. So if you have a longitudinal study, then the session could indicate which time point it is. And then your file looks like this. So if you have a T1 weighted file, then we have subject 1, the session 1, you know the modality, T1 weighted, and you know the extension. Okay, I see the colors have shifted somehow, but yeah, you'll get it. And then metadata is also stored alongside the image. We have subject, session, and a JSON format for this image, where we can store all the metadata, which repetition time, which echo time, which contrast, and all the things that are kind of flexible parameters can be stored there. This looks like this then in the end. So this could be a raw DICOM data file. So you have no idea what this is if you haven't created it. And the bits data structure looks like this. There's a participants file listing the participants. Then you have subfolder for the different subjects. And then you see anatomical imaging, so the T1 image. Then you have time series of resting state, fMRI, and diffusion weighted images for the subject. And if you have run pre-processing or analysis, then we would get another folder inside the subject one folder with derivative data. And if you now get this data set, then you can decide, oh, do I want to use the derivatives? Do I want the pre-processed data? Then we can go directly to the folder. Or if you want to use the raw data for your own pre-processing because you think, I'm not quite convinced of the pre-processing pipeline of my colleagues, so I will do it on my own. And you can compare that afterwards. And you can compare the images, and then you might realize that you have done something wrong, which the others have already corrected for, or something like that. So this is pretty cool that everything is stored in one file. So it gets a bit bumpy, because then if you have more things defined, then you have pretty long names, but they make sense, and all key values are like with dashes, and the entities are separated with underscores. So you get a quite nice overview. Okay, it already took really long, so I don't know what time is it? Okay, I think we can make a 5-minute break. Would that be okay for you? And I'll switch on the title for slide to create a thumbnail for the YouTube video. So let's meet. Okay, let's take a 5-minute break and take time for questions, for sure. Okay, there's one. Could you briefly explain again what is meant by the orientation isotropic or versus anisotropic of a voxel? Okay. Let me switch to the slide again. So it's, if you have a voxel that has the same size in any dimension, then it has not a direction. But if you have a voxel that has a greater size in one dimension, then the voxel clearly points towards it. So the resolution is different if you look, for example, to a sagittal slide, or if you look to a coronal slide. This is because of the voxel sizes are different. So the signal inside one voxel gets averaged, because that's the smallest part we can look at in an image, and so we have an average across one dimension that averages more information than in the other dimensions. And this is done in clinical practice, because then the time for acquisition is reduced. And if you need only, like, if you know what to look at and where to look at, you can then say, okay, I can use this trade-off with one dimension being with a lower resolution, because then the scanning takes not this long, and you know what you want to. And the contrast can be increased in the dimension as well. Yes, I hope this kind of explains it. If it doesn't, okay, thank you. Any other questions by now? Okay, then I would say 1 more minute, like till 5:55, and then we're gonna start with the analysis or the acquisitions of the different modalities. And I think we are definitely not going through all of the slides, and maybe I'll show you some of the slides then in the next session. Yeah, but I hope that yeah, it was not obvious to all of you what I've said before. You can give some feedback if it's too like shallow and too superficial, the introduction, then I can speed up a bit. But I don't know how the expertise of you is. Maybe we can, like in the last 30 seconds of this little tiny break, we can quickly make a little survey. Just post in the chat if you ever have worked with or an imaging file, or yeah, and in any case, if yes, please, yeah, just write down the analysis method or your experience with it. This would be super nice. So please write just into the chat 2 or 3 words, if whether you have ever worked with a neuroimaging data, or, and in the case of yes, what have you done? And then if the case nobody answers, then I suppose I stay with the more in-depth and general. Okay, let's go on. So for structural MRI, just a quick summary. So we there's an application of a strong magnetic field, the B0, which aligns hydrogen nuclei. This excitation pulse, the radio frequency pulse, tilts nuclei from the alignment. The nuclei precess back to the alignment once the pulse is switched off, and so they emit a signal. There are longitudinal in the Z direction and transverse signal components, and the realignment in the different components can be measured. So the time constant for the longitudinal realignment is T1, and the time constant for the transversal realignment is T2. So as you can see, you have a transversal alignment, transversal magnetization, and once the pulse is switched off, then the proton goes back, aligning to the B0 magnetic field. This can be also seen here. So for the T1 relaxation, we have this back to this precession progress back to the magnetization, and the T2 transversal magnetization is decaying. And based on like the percentages, we can make this time constant and can measure what the T2 and the T1 time constants are for different tissues. So yeah, as I said, there's a contrast between the different specific in T1 and T2 relaxation times. So the final contrast is made after an excitation pulse, and then we're listening after a certain echo time, and then we're doing the same thing again, defined with the repetition time. And based on like the repetition and echo time, we can have different weighted images. For a short echo time, a short repetition time, we have a T1 weighted image. And for a long repetition time and a long echo time, we have a T2 weighted image. And this is only, those parameters are set to increase or maximize the contrast. So we have a relative measure of differences in magnetic signal for different tissues. So maybe let's try it again and make another quiz. So what are the most basic pulse sequence parameters that impact T1 weighted and T2 weighted image contrasts? And which one is larger? Okay. And the second question would be in the T1 weighted image. Which one is brighter? White matter, gray matter, or cerebral spinal fluid? And I'll answer it myself. So the repetition time are the parameters, and the echo time. So those are two pulse sequence parameters that dictate the image contrasts. And the repetition time is, of course, longer than the echo time, because you first gotta listen to the signal, and then you have to decide excite the protons again. And yeah, the white matter track is brighter in T1 weighted images. Here's the solution. So we have a T1 weighted image and the T2 weighted image, and you see the different tissue types. So gray matter, white matter, cerebral spinal fluid, or bone. And you see the different relaxation properties for the different tissues. And as you can see, at T1, gray matter has less signal than white matter. So gray matter is a darker shade than white matter. It's the other way around with the T2. So gray matter has a higher signal at the relaxation time T2 as white matter. And there we have the contrast between gray and white matter. And we can see here like kind of a switched or an inverse negative picture. Okay, for T2 weighted images, we can quantify anatomy. So we can pretty nicely see structural volumes. And in T2 weighted images, the cerebral spinal fluid is light, the white matter is dark. And yeah, we can use this to identify lesions or tumors, or to spice up or make pre-processing more precise. Then we have cerebral spinal fluid, we have a proton density. There we can identify demyelination. And we have flare, which is called fluid attenuated inversion recovery. And this is, there it's similar to a T2 weighted image, but the liquids are suppressed. So we don't care about cerebral spinal fluid, for example, and we can see only matter, gray matter and white matter. Okay, so but to kind of condense it, what do we want? We want a high spatial resolution to take a close look into the brain and to differentiate between brain areas which are pretty small. We need a low scan time because we can't put, for example, other patients or individuals can't put them into a scanner for hours. It's pretty close and pretty narrow, and so we need a kind of ethical scan time where we don't torture the participants. and we need a high image contrast, so a high signal to noise ratio or contrast to noise ratio in this case. But there are certain trade-offs. So we can control the magnetic strength, which increases the signal to noise ratio. So if we have a 3 Tesla scanner and compared to 1 Tesla scanner, the signal to noise ratio is increased. If we have a 7 Tesla scanner, it's even better. Then we have different sequences. So a magnetization prepared rapid radio echo image MPRAGE, which is commonly used in neuroimaging. And this is based on gradient echo, a different sequence, how the signal is how we listen to a signal. And yeah, there's a gradient then introduced after a radio frequency pulse. And with that, we can accelerate the image processing, the image acquisition time. Yeah, so this can be done, but this comes with an cost of more noise. If something is more, there has to be some trade-off somewhere. And the high image contrast, another problem is proportional to the voxel volume, because if you have a signal that comes from a voxel that has the dimensions of 4 millimeters, then there's much more signal than a voxel that has only 1 millimeter. So the image contrast or the signal to noise ratio is increased with greater voxel volumes, and it's also proportional to acquisition time, the square root of the acquisition time. So if you take more time and average across more space, then you get a better signal, and so the signal to noise ratio is better. So there's a trade-off with resolution here as well. So if you want a high resolution and a high contrast, you definitely have to increase the magnetic strength. Otherwise, it gets difficult. Okay, then there's a thing regarding resolution, because most of the brain structures are smaller than the resolution. So we can't differentiate between brain structures, and this is called partial volume effect. So we have resolution usually about 1 to 3 millimeters, but we have brain structures that are sufficient or are even smaller. So it could be that portions of different tissues are averaged together in a slice. So this schematic here is trying to show that. So we have a 4 by 4 millimeter voxel, and you have gray matter and white matter. And yeah, the mixture of the signal for both tissues is then the signal. So it's a weighted average. And the weighting is based on the proportion of the volume or the fraction between the signal of the gray matter and the white matter inside this voxel. So if you increase resolution, so decrease voxel size, you see what happens. You're averaging not over the whole volume here. So you have voxels that only emit the signal for one specific tissue type. Yeah, but we can't go down with the resolution like I showed you with a micrometer scale with a big brain, because those were invasive slices. So we have to deal with partial volume effects even with 1 millimeter by 1 millimeter voxel size. So this is called partial volume effect, and we always have to keep this in mind. To show this in an image, this is pretty interesting, because intuitively you would have thought that this picture looks nicer, so this has to be a better resolution. But the cause of this grainier looking picture is that if we just reduce increase the resolution, we get more noise, so it looks grainier. But we can clearly more define the barriers between gray matter and white matter. And this is this because of the partial volume effect is especially in those barriers a bit problematic, and it works not so good. So if we increase the resolution, we can differentiate tissue types better. So okay. But those are not only the problems of MR imaging. We have also kind of technical and physical problems that was also called artifacts. And yeah, causes could be scanner hardware, equipment, an error of the operator, subject movement, physiological confounds. So basically, in each step, problems could occur. Yeah, and we for some artifacts we can control some artifacts. For others, we have to really observe the image. If they are present, then we have to optimally we have to redo the scan. So looking at the data at the first point when directly after acquisition is mainly a good idea, because then there's the possibility to rescan and without the artifact or without the problem. So there are motion artifacts. They occur because the subject is moving in the scanner. Yeah, this is extremely common. Yeah, this can be reduced if you ask the subjects to remain still or make it more comfortable in the scanner with cushions or yeah, for example, or yeah, if you see those artifacts, yeah, you have to rescan or to exclude the participant, because movement biases the analysis and yeah, decreases the gray matter volume without and therefore we have to be careful with analysis. Okay, then there is bias field inhomogeneity. This is based on imperfections in those radio frequency coils. They are mostly present and noticeable by eye, and they can be well corrected by those standard analysis methods. So this is an artifact that can be corrected with pre-processing. Then there's ghosting. This is based on complex reasons and it is extremely common, but yeah, usually very weak. And yeah, you can try to reconstruct and extract the brain, so you ignore the ghosting. Then there is a gradient nonlinearity distortion. This is also based on standard like imperfections of gradient coils. And yeah, because this is more noticeable in the neck as compared to the brain, you could ignore it if yeah, and you can also reconstruct it with a so-called gradient nonlinearity correction. What also helps is if you check that the brain is really in the center of the magnet. Because if the brain is kind of displaced, then this nonlinearity distortion can occur. Okay, then there's a wraparound. This is mainly based on a field of view that is set to small. So yeah, it's cut here. The brain is cut here, and yeah, if the wraparound does not go to the brain, then it can be ignored. Otherwise, yeah, it has to be scanned another time. Then there are physiological confounds, which you can't really switch off. So heartbeat or blood flow of the participant, this is periodic. Yeah, this one thing is you can do this with a flare image. So you nulling the liquids, so the blood is nulled, or you can sorry, or you can measure physiological, or you can do physiological measuring. So you can in parallel measure the heartbeat and use this then for correction afterwards in the processing step. Yes, this is artifacts. Now we can go to the summary of structural MRI. There are varieties of images with different aspects of anatomy. The T1 weighted images are most common. They have a good contrast to noise ratio and tissue discrimination. Then the isotopic acquisitions are better for neuroimaging research. The image intensity depends on the proton density and T1 and T2 relaxation times. We talked about this as well in detail. The structural MRI is not quantitative, so the intensities are representing main tissue types in the brain, but not how many tissue type in the brain is. So yeah, there are many different artifacts. So always check your data, please. And yeah, artifact correction is in some cases possible, in some cases it is not. But if you know how the artifacts look like, you can decide then if you can keep the image or if you have to discard the image. Okay. What time is it? Can't see my clock somehow. Okay, 20 minutes. So maybe we will get it to through the main modalities, and yeah, the analysis pipelines we can do then next session, which is pretty cool because then I will present one major analysis pipeline. So we can first get an overview of existing pipelines or of general pipelines, and then see specific examples. This is which is really useful and helpful. So okay, diffusion MRI. It is a measurement of Brownian motion, basically. So if you put ink into the water, it equally spreads out. This is called diffusion. And a process because of biological restrictions of an axon, so often the diffusion is altered by axons. The water molecules cannot pass the cell membrane because they are lipid, and so water molecules inside a cell or between axons that go the same direction can only diffuse in the direction of the elongated axons. And the diffusion MRI analysis quantifies the physical properties of the directional diffusion. One model to quantify this diffusion is the diffusion tensor imaging. So diffusion MRI measures diffusion of water molecules, and one model to model like white matter tracts is diffusion tensor imaging to model the direction of diffusion. Yeah, and we can derive the mean diffusivity and fractional anisotropy from the diffusion MRI signal. Yeah, the fractional anisotropy is depicting how different the diffusion is in all directions. So a high fractional anisotropy means that the diffusion is in one preferred direction. So we have, for example, we have an axon that points in one direction, so the diffusion is bigger in one direction than in the other. Okay, bigger in one than in the other. So this can be then represented with a diffusion tensor. This is then a quantitative analysis of the magnitude and directionality of the diffusion of water molecules. And if we have like diffusion that is equally in all directions, so everywhere where no axons are, so water can pass everywhere, we have an isotropic diffusion, as you can remember with the voxels. So the eigenvectors or eigenvalues of the vectors are all the same, are equal. And if there's diffusion in a preferred direction, we have an anisotropic tensor that points towards the direction of diffusion. This can be then used to measure the diffusion in the brain and to reconstruct anatomical connectivity. This is based on the direction of axonal fiber bundles in each location. So you estimate basically for each voxel in the brain So each voxel, you estimate the direction of diffusion in each voxel. And if you trace down the directions from one gray matter voxel to another one, you can trace the white matter tracts between those regions, voxel by voxel, and this is called tractography. Okay, for this, there's a special acquisition. So we have to set special gradient fields that sensitize the image to diffusion in particular and to many directions. So this can be showed here in a sphere, in a three-dimensional. So in the center, the center here, and all the dots are showing the different directions of gradient fields that measure the diffusion in this specific direction compared to the center. This requires a set of many 3D images for each diffusion direction. So we need this kind of equiplanar imaging I told you. So we have a fast image with like 3 to 4 seconds per image, which leads then to a total image acquisition time of 5 to 15 minutes. So we have a fast sequence, but a limited resolution, and it's kind of sensitive to geometric distortions. So especially for diffusion MRI, it's to keep in mind. What comes out are 3 diffusion weighted images, or like not 3, but 30 to 200 images, each for each direction defined in the sphere. But to simplify it, we have a diffusion along like 3 axes shown here, and you see the signal strength for diffusion in this direction. So in the x-axis, you see here more intense diffusion than in the y and z-axis. So this diffusion weighted image for each direction can with that we can infer the diffusivity in a specific direction. Okay, and to model then white matter tracts, we can describe this diffusion with a tensor model. So we can model the diffusion characteristics within a voxel. And those tensors are represented by ellipsoids, so 3D ellipses, characterized by eigenvectors that have eigenvalues. So the eigenvalues are lambda. And yes, I won't go into detail, I think. So the tensor model consists out of a mathematical model. So G is the unit vector, B are the parameters of the measurement, then S, the functions, is the diffusion weighted signal, and S0 is the signal conducted in a measurement without diffusion weighted imaging. So this is a baseline basically. And then we have a matrix, a diffusion matrix. This is kind of a variance covariance matrix of diffusivity along the 3 spatial dimensions. And we have then a model with 6 free parameters to fit the data to. And there we get then the eigenvalues of all the directions in 3D space in each voxel that describe diffusion. So if we see if this is anisotropic, and if you see that we have this vector is bigger than the other 2, then we have preferred diffusion preferred in this direction. This can be described with fractional anisotropy, as I told you before, and this is basically a normalized variance of the eigenvalues of the tensor. So it looks a bit complicated, but it's just the eigenvalues of the tensor, and you can characterize the degree in which the distribution of this diffusion is directional. So if you have high fractional anisotropy, the diffusion is preferred towards one direction, and if you have a fractional anisotropy of 0, then there's diffusion in each direction equally. Okay, as you see, if we have a voxel with a high FA, we can infer that this voxel is probably white matter tract or something, and we can trace those tensors then along. Okay. So if we see this in an image, the high fractional anisotropy means that there is a fiber population with coherent orientation. So see a high signal in this FA image, you see all those white matter tracks that have a specific orientation. Medium to low FA value means that there are voxels with fiber crossings. So there is a preferred direction, but yeah, it's not quite clear which it is. And lowest FA, because 0 means that there is no white matter tissue, so the diffusion of water molecules can go in all directions equally. Okay, then we can also picture mean diffusivity. This is just the degree of diffusion, which is independent of direction. From that, we can infer then the diffusion rate along the primary axis, which is called axial diffusivity, and the diffusion along the minor axis. So the axial diffusivity is the eigenvalue of the vector in the preferred direction, and the average diffusivity is the other one. So those measures can be used to further characterize the tensor. And this can be used then to color code the diffusion orientation if you overlay the diffusion with a fractional anisotropy map. And then we have quite nice and this kind of fancy visualization we see everywhere. So if there's a magazine saying something about the brain, they probably print the diffusion weighted images on top because they look so nice. And yeah, this is how it's done. And we have one tensor at each voxel indicating the direction. So blue for superior, inferior, red for left, right, green for anterior, posterior. And yeah, we can overlay all three to have this nice color-coded image. By that, we can do tractography. So we can trace along the voxels with the directions and connect gray matter structures with that. So I won't go into detail, but there are different white matter tracks we can reconstruct based on this kind of diffusion tensor imaging. We can also then kind of make a more comprehensive structural connectivity. So we can connect all gray matter structures with the white matter tracks to define connectivity-based regions. So as a summary, diffusion of water provides an indirect information about those axonal fiber directions. There's a quantitative measurements of mean diffusivity, and yeah, the anatomical connectivity via tractography is not quantitative. There are many images in different directions needed for the data set. So in the sphere, and yeah, we need a fast imaging sequence, this echoplanar imaging. So we have a limited resolution and signal to noise ratio, but we can acquire a baseline image that is not diffusion encoding and calculate the diffusion tensors which lead us to a tractography, which can be modeled with diffusion tensor imaging as a mathematical model. Okay, I think the time is over, and I will, maybe we take the time for a few questions now, and we're gonna continue with the last slides then in the next session, because it would be a pity if we rush through those slides, I think, and I guess it was already a lot. So please go for any questions if you have one.